Okay, it is 10:31, so as people filter in, but we can let them filter in, but we'll get started. So hello everyone and welcome to the 46th Great Lakes Phragmites Collaborative webinar. And this is our first webinar of the 2022 series. So I'm Teresa Greninger and I'm joined by my colleague Sam Tank from the Great Lakes Commission, and we are hosting the webinar today on behalf of the Great Lakes Phragmites Collaborative. Our presenter is Dr. J Mctavish with the Smith Forest Health Lab at the University of Toronto Ducks Unlimited, Canada. His presentation today is entitled A New Tool in the Toolbox, an update on biocontrol of introduced Phragmites. Before we get to our featured presentation, I wanted to introduce the Great Lakes Phragmites Collaborative for those of you who may not be familiar. The Great Lakes Phragmites Collaborative is a partnership to link people, information, and action on invasive Phragmites. The collaborative is coordinated jointly by the Great Lakes Commission and USGS Great Lakes Science Center with funding from the Great Lakes Restoration Initiative. The collaborative facilitates communication among stakeholders across the region and assists in development of management tools and programs that serve as a resource center for information on Phragmites biology, management, and research. The collaborative is led by a multi-agency advisory committee and applies principles of collective impact to advance common agenda for Phragmites management in the Great Lakes region. The Great Lakes Phragmites Collaborative is working towards advancing member strategies that are outlined in our common agenda. The common agenda is a big step towards aligning our regional goals and to establish a shared vision for change. The purpose of the common agenda is to clearly define the intentions for the collaborative, develop a shared understanding of gaps in the problem areas surrounding Phragmites, and lastly, to highlight the areas and strategies that we are working on or wish to work on. And you can learn more about our regional efforts and how you can get involved at our website. So a big component of our work and one of the ways we engage with Phragmites managers is the Phragmites Adaptive Management Framework, or PAMF. PAMF utilizes participatory science to provide data-driven Phragmites management guidance through an adaptive management framework. What that means is that we have established a way to learn from what Phragmites managers are doing and turn that data into annual guidance to help managers make effective and efficient management decisions. So we encourage anyone currently watching this, if you're managing Phragmites, to consider joining PAMF. And you can see the information on the slide. And anyone interested in joining PAMF or taking advantage of PAMF's guidance can enroll in our free virtual training course by following the URL on the screen. We have a lot of training resources that help you get started and are always available to answer any questions at pamf@glc.org. So if you're interested in learning more about Phragmites management and research, you can check out our resources on the website, which include management guidance, recordings of past webinars. You can also join our listserv to connect with hundreds of other professionals or sign up for our bi-weekly newsletter, which includes quarterly research roundup of academic papers published in the previous months. You can find links to join the newsletter and the listserv on the front page of our website at www.greatlakesphragmites.net. And today's webinar will be recorded and uploaded to our YouTube channel and website. So you can access an archive of all of our past presentations. You can also view and register for upcoming webinar and presentations by following the URL listed here. So hello to those who are watching the recording from the past. Please share with your colleagues and don't hesitate to reach out, even if you're watching this long before the presentation date. And of course, connect with us on social media, tag us in your Twitter, Instagram, and Facebook posts, all at GLFrag. And while we always strive to minimize technological issues during these webinars, we apologize for any issues that may arise. Further, I'd like to cover a few logistical items before we get to our presentation. So first, audio for this webinar is automatically streamed through your computer speakers unless you're using the call connection sent out in the register confirmation email. In order to minimize the potential for interruptions and background noise, we have automatically muted all attendees. Attendees will remain muted throughout the webinar. And to save bandwidth, we ask that all attendees please turn off your cameras. And we do encourage and will accept written questions from attendees during the webinar. If you have questions, you may type them into the chat box, signified by the chat bubble icon at the top of the webinar screen. And questions may be submitted at any time during the webinar. We'll do our best to answer all questions and comments through the webinar, but if you need to leave early and we haven't gotten to your question yet, we encourage you to look back at the webinar recording to see your question being answered. If you're expecting any technical difficulties, you may send a message through the chat box, as well as you can direct that message to the host or email us at phragmites@glc.org. And so all that, now it is my pleasure to introduce our presenter. Our presenter today is Dr. Michael J Mctavish. He is a Mitex Elevate postdoctoral research fellow working with the Smith Forest Health Lab at the University of Toronto and Ducks Unlimited Canada. His current research includes biological control of non-native invasive plants, including Phragmites and garlic mustard, and the ecology of non-native earthworms. So thank you so much for sharing your work with us today. I will transfer the presentation control over to you, and you can start and take it away.
Great, thanks very much. I'm gonna pull up my slides here. Let's make sure we got that working all right. Everything's looking okay?
Looks good.
Yep, perfect. All right, so good morning, everyone, and thank you very much for joining us here. I hear we've got a good turnout and probably folks from a number of different places, which is really great to see everyone coming together for this. A special thank you from my end to the collaborative for the invitation to do this talk today. Excited to share some of what we've been doing here. So as was mentioned, I'm going to be providing a bit of an update on our pilot program for biological control of introduced Phragmites, which is taking place in Ontario, Canada right now. Phragmites is probably a problem that many of us are familiar with, so I'm not going to dwell on this too greatly. But what we're looking at here is introduced Phragmites, and that's how I'll typically refer to it. Specifically, we're talking about Phragmites australis, which is a subspecies of that species, and it's considered one of the most invasive plants in North America. We believe this came over from Eurasia in the late 19th century, and of course it causes all manner of different problems. Generally, it forms these very dense monospecific stands that can reduce biodiversity, alter various ecological processes, increase fire risk, reduce property value, and on and on. We mentioned this is a subspecies. We do also have a native subspecies as well, Phragmites australis americanus, and it is also threatened by the introduced subspecies through competitive displacement and also hybridization. So we do have various controls to help manage Phragmites, and under the right circumstances, these can be quite effective. But of course, Phragmites occurs in a number of different contexts, and so in certain cases, these options that we have can vary in their efficacy, affordability, feasibility, how practical they are. And so with this kind of problem that is so large and so challenging, what we've really needed for a long time are additional options, always additional options to supplement the tools that we already have. And one of those new tools that we'd like to bring forward and share today is biological control. And an important theme throughout all of this is really we're not talking about a replacement tool, but a supplemental tool. So we like to think of it as giving people another option in this toolbox to help deal with the challenge of introduced Phragmites. So what is biological control? Biological control, or biocontrol, is the use of live organisms to suppress an introduced pest in the broadest sense. Why is this an appealing tool in general? Many weeds and pests in general can do quite well because they manage to evade natural enemies. They sort of leave these behind in their native range, and these are populations that would normally help keep their numbers in check. So biological control is about reuniting the pest with some of these natural enemies to help manage their population. These natural enemies are selected to be highly host specific. So they go after this plant and this plant alone, which means that they are very target specific and have low environmental impact on, for example, other species or other abiotic aspects of the surrounding environment. Biocontrol has a lot of appeal because it can be self-sustaining. So ideally, once you get it going, organisms are reproducing on their own, and they track the host through both space and time as the populations reproduce and disperse. Because of that self-sustaining nature, biocontrol can also be highly cost-effective, particularly at large spatial and temporal scales where other methods may become prohibitively expensive or just logistically too demanding to be practical. Biocontrol is sometimes thought of as a bit of a new tool, but it at the same time has a fairly long history in Canada. So since the first programs in 1951, a total of 81 foreign insects have been released against 29 different weeds. And this long history is well documented, in particular through a series of books that are published through CAB or later CABI. So this is the Commonwealth Agricultural Bureau, and then the international re-implementation of that. There's actually a sixth publication in this series that is currently in preparation that we're contributing to at this moment. So this isn't a brand new tool. The general goal of biological control, we can sort of depict this here with a sort of a general case. If we look at our dotted red line there and we say this is sort of the population density of a weed like Phragmites, and then the density of our agent that we're releasing is in the solid blue line, and then we have time along the horizontal axis here. We can sort of break this down into a few different phases to show the situation that we're trying to achieve. So basically, you start off with a situation where you have a problem weed that exists at very high population densities. You release a fairly small population of the biocontrol agents early on that begin to establish and start to reproduce. You then enter a second phase where over time, ideally that agent population grows in size, and as they do so, they put increasing demographic pressure on the weed, and the weed population density comes down. And then you settle on sort of a longer time sort of balance where you have a fairly low density of the weed, but not zero, which is sort of an important aspect to consider here is that we're not talking about complete eradication, we're talking about suppression to low levels. So you reach this state where the weed and the agent sort of coexist at fairly low densities. So that's kind of what we're trying to achieve here. And another thing to draw attention to is the timeline on all of this. So if these are years, this is not a process that happens immediately. Again, this is sort of a longer term solution for a large scale problem. So these goals are realized over years. Biological control programs take a lot of work and a lot of time to put together and to put onto the ground. So I'm going to break this process down for those who may not be as familiar with it, sort of as it exists largely in Canada. This can be organized into sort of 4 larger stages or 7 discrete steps, kind of different ways you can break it down. So these programs typically start with this stage of horizon scanning and sort of foresight and essentially planning. So the first thing that you need to do is understand the species interactions that are occurring in the invaded range, in this case in Canada. So this would be, for example, looking at what introduced Phragmites is doing, its impacts, how it occurs, how it coexists with things like native Phragmites, and so on. The second stage deals with foreign exploration for agents. So you're trying to identify suitable candidates to use as biocontrol agents. In a classical biological control program like this, you're drawing agents from the native range of the species. So if you're in North America, you would be doing overseas explorations to try and identify agents in the country of origin that are going to be specific and that are going to have an appreciable impact on the weed that you're trying to target. Once you have that, so a candidate or a few candidates, you move on to a stage of risk assessment and release. And there's sort of a lot packed into this particular stage. Once you have those candidates, you want to first look at their biology, so in the field and under experimental conditions, and a large amount of time is then devoted to host range studies. So this is a period of testing using a number of different approaches to test and ensure that the candidates are specific to the weed that you are targeting. So they're going to damage that and nothing else. Once that is then lined up, you move on to the creation and submission of a petition for agent release that is based on those data that have been collected to date. And then if that is approved, you go on to a stage of actually rearing biocontrol agents and then releasing them into the field. Once they're out in the environment, you move on to the final stage of post-release monitoring and evaluation. So once they're out there, you're trying to monitor for establishment to see that you have a population that has taken hold, and impact assessment to see what they're actually doing to the target weed and make sure that there aren't any other non-target impacts. Once you know that they're there and they're sort of doing their thing, you have this final step of agent redistribution and long-term assessment. So you know how to get them out there, you know what they do. Now you're just focused on distributing them across a larger area and assessing their impact and spread over a longer term period. And we're going to get to this a little bit more in another timeline, but while we're on this breakdown of it, I'll also just flag that when we're talking about this Introduce Phragmites biocontrol project, we are essentially at this third stage and fifth step of rearing and field release. So that's where we are in this breakdown of the process. So an important thing to flag at this point, and I think it's relevant, really relevant for this group that we're meeting with today, is to recognize that Introduced Phragmites is really an international problem. It exists on an international widespread scale. So this is pulled from the USDA plants database. In the sort of kind of light blue coloration there, we see states and provinces that have the introduced subspecies of Phragmites, and we see that it is widespread and exists on both sides of this border. So this is an international problem, and as such, what it really needs is an international solution. So developing this biocontrol has been an international multi-stakeholder solution that's really been in the works for now over 2 decades. I haven't been personally involved with it for the duration of that, but there are people who have been. So I'm not going to go through all of these here, but we have pulled out some key personnel who have been, especially with this pretty much from the beginning, various institutions and organizations in the US, in Canada, and in Europe who have collectively come together to make this happen. So the important takeaway from this is really that it takes an army and it takes an international army to get this done. So what has that army actually been working on? So here's a timeline of this Phragmites biocontrol program. So as I mentioned, really this goes back at least 2 decades. So those initial proposals and insect exploration, so those early stages and steps of that process, they began in 1998. By 2005, there was a short list of host agents that were candidates to be used for biocontrol. By 2007, these had been refined to 2 species shown here, and I'll introduce them in a couple of slides. But this is where we hit that really extensive and really important period of host range testing. So again, these are various experimental tests to ensure that the agents that we're looking at are highly specific to the target weed. So that went on for a number of years through a number of different institutions, and finally culminated in a petition to release these 2 agents against Phragmites in Canada and the US that was submitted in 2018. This was approved in Canada by the Canadian Food Inspection Agency in 2019. In the US, the petition is still under evaluation. It's a different process that generally involves a larger number of steps, so it's still moving through that regulatory process at this time. And then that brings us to the present day Ontario pilot program. So here are those agents that were mentioned on that timeline. Biological control of Phragmites uses 2 European moths with stem-boring larvae. So shown here, we have Archanaraneurica and Lenisageminipuncta. And I'll note that in some older documentation, you'll see Lenisageminipuncta categorized within the genus Archanara, but it is the same species. So these are fairly small, somewhat drab kind of moths, nothing special to look at. But the actual damage that we're looking for comes from the larvae. So the larvae will mine shoots of introduced Phragmites, and indeed the larvae will move through several shoots over their life cycle as they develop. So they're able to cause quite a bit of damage by moving through several shoots per larvae. And that damage will cause, depending on the stage and state of the stem, either wilt or potentially mortality, reductions in reproductive output, and various other things that overall will reduce the population density of the Phragmites and reduce its ability to spread and its ability to compete. So in these photos here, we can see sort of on the right, sort of the leftmost panel there, this is a stem that has a larval borehole from one of these larvae. So this would be them entering or exiting the stem. That would probably be from a later instar larva because it's fairly large. And then on the very right, we can see a larva of Lenisageminipuncta. This would be a fairly late instar larva. This is what you might see if you were to crack open one of these infested stems, and you can see them chewing their way through the pith of the stem there. The life cycles of these two species are slightly different, but overall fairly similar. Archanaraneurica flies a few weeks earlier in the season than Geminipuncta, but otherwise they're quite similar. So if we go through a quick breakdown of what that life cycle looks like, we can start with the larvae, which again is the life stage that actually does the damage to introduced Phragmites that we're interested in. So the larvae will hatch in the spring, and they will feed in the shoots for a period of about 4 to 6 weeks. And again, as they develop through their various molts, they will switch between stems, and so at large population densities can target a large number of stems in a stand. They will then pupate in the stems themselves, actually, for a period of about a month, give or take. And then they emerge as the fairly short-lived adult moths. They will fly, ideally mate, and then lay eggs. So the females can lay approximately 150 eggs, and these are sort of glued underneath the tight-fitting leaf sheath of the plant. So they're sort of between the leaf sheath and the stem, kind of protected, and they will sit there until the following spring and overwinter in that location. So what I want to do now is provide a bit of a research update on the work that has happened from 2019 to present. So 2019 is coincidentally when I became involved in the project and close to when we were able to begin releasing agents in Canada. So this pilot program has been based in Ontario and is built around 3 primary research objectives. And these in some ways mirror many of those stages of that biocontrol program that we went through earlier. So it's important to understand the system that you're working with and what you're targeting. So one of the first objectives here is to conduct background monitoring and investigation of Phragmites, so both the native and introduced subspecies, again, to understand this context that we're working in. Really, the meat of this is, of course, to develop, to test, and compare different methods for how we rear the insects, for how we release them into the environment, and then how we monitor to see whether they've established, how they spread, and the impacts that they have. And finally, we're also, while we're doing this, trying to actually get insects onto the landscape and establish these nurse sites. So these would be locations where populations are established and reproducing, sort of where they've kind of settled in, if you will. And these are really useful to us because they can be examples of long-term populations and also sources of insects and eggs or whatever we're harvesting to further additional research and rearing. So if we take some of this current research and we drop this onto a timeline, again, sort of from 2019 to present, when I started with this project, we began with a background survey of Phragmites. So again, both the introduced and native subspecies across a fairly sizable latitudinal gradient in southern Ontario. So you can see Lake Erie in the bottom left of the map there, and then we're going up the St. Lawrence River towards Ottawa. We did sort of 50 to 60 sites that we were looking at from historical mapping data. And we had a few objectives with this. We were trying to identify locations where we could do potential biocontrol releases and experimental work in the future. And we were also creating baseline data for how these populations behave in absence of biological control, and also furthering our ability and tools to help identify between these subspecies. There is sort of, we know from host range testing that we can sort of talk about later as well, that they have a very strong preference for the introduced subspecies. And this is just something we want to make sure we confirm in the field. So we've been trying to identify locations where these two subspecies will co-occur. So while we've been doing that, we're also developing using a combination of molecular and morphological tools, sort of basically guides to discriminate between these and to screen for potential hybrids in the field. In 2020, this was sort of the first time I was involved with any releases. One of the big questions through all of this is what life stage you release as. So you have a few options in this case. You can release as eggs, you can release as larvae, as pupae, or as adult moths. And there are various trade-offs to each of these different methods. And even within each life stage, you can have different logistics that you use to try and get them into the field. So in 2020, we started with pilot releases of eggs. Eggs are probably one of the easiest life stages to work with, and just because you're not dealing with sort of a mobile organism at that point. So the first thing that we tried is depicted on the left there in that photo. We tried to emulate the natural egg-laying behavior of the species by gluing groups of eggs to these pieces of index card that were then secured to Phragmites stems in the field. And we thought this was sort of a good way to try this. What we found very early on, however, was that these eggs would encounter very high predation. So if they were sitting in the field for any period of time, we'd get probably, we're suspecting ants and spiders and various other things sort of ripping the eggs off and consuming them. So we tried to release at a second site where we compared this open card method with something to give the eggs a little bit more protection. So we created this screened in cup. So the eggs are actually inside the cup there, and it's got a window screen mesh bottom on it. The idea being that the mesh is sized such that the larvae can make their way out of the mesh, but will be protected from larger predators. And we did find that this was able to protect a large number of the eggs, which was really useful to learn. This last year, 2021, we were able to get quite a bit done in terms of releases. And as, of course, we're all probably aware, this has been a challenging time to do a lot of this work in terms of pandemic and restrictions and so on. So we were really glad that we were able to do what we were able to do this past year in particular. This was a good opportunity to release at a number of different sites and test out a number of different methods. One of the first things we looked at this year was the question of overwintering storage. So you're trying to figure out what life stage to release as, how to do that, but also how to store. So no matter how you're releasing them, eggs need to go through that winter period as eggs, and you need to figure out a way to do that that will maximize the hatch that you get from that. So we did some tests with the eggs to see sort of the best conditions for doing that. And we found that exposure to fluctuating outdoor temperatures seems to be a good way to achieve very high hatch, which was useful to know. For one of our species, Archanaraneurica, we're also able to rear them on an artificial diet. So if you look in the sort of middle left, you'll see some little cups there, and each of these has a larva of the species, and you see those sort of pale cubes. That's an artificial insect diet, and they're able to complete their development from egg to pupae. In this case, we usually take them completely within a lab environment. So this is very useful for us to be able to produce insects sort of indoors in a very sort of standardized environment. And this can allow us to produce pupa or moths or eggs or whatever life stage we'd like. So we did some rearing with this one species for that. A lot of our releases this past year actually went out as larvae. And we used two main methods to do that. One was with just loose larvae, and this was largely a response to sort of the glut of hatch that you get initially when you take these eggs out of storage. You get thousands of larvae potentially hatching on you, and it's sort of a lot to manage. So one tool that we wanted to explore was the viability of essentially just putting those larvae in a container and sort of dumping them into a patch in the field. So that's one thing that we tried as sort of our low-tech method. The other thing that we've had a lot of success with, and I'll show a breakdown of what this looks like later, is to inoculate the larvae into stems. So if you look in the bottom photo there, you can see there's a block of about 50 stems. So these are Phragmites stems about 30 centimeters tall that have been cut and then inserted into this piece of moist florist foam to keep them green for a little while. So what we do is we actually take these stems from the field, we'll cut the bottom off to expose a lower internode, and then inoculate. We're doing about 3 larvae into a given stem. You plug it back up, it goes into this block of stems, and they'll happily eat away inside of that stem in a nice little protected environment, and that'll keep them busy for about a week. And then you can take out that whole block of stems and just put that out into the field. So once they're done eating, they will then ideally make their way out of that stem and crawl away into a new stem in the population, and now they're on site. So this has been a really great method. It is labor intensive, so we're also looking for alternatives, but it does seem to be a good way to do this. We've also been experimenting with pupil releases as well, and this is kind of a nice way to do it because with larvae or with eggs, you can get very high mortality at these earlier life stages. You certainly can still get mortality later on, but you sort of know they've at least made it this far in their life cycle, so you know how many you're putting out. So we've tried pupil releases. Typically what we do sort of in the top right and middle right, we'll have these containers that are open to the environment. They've got just some vermiculite in the bottom to keep things moist, and you put some known number of pupae in there. These can go out into the field. The pupae, adult moths will emerge, fly out, ideally mate, and lay eggs in the patch where you've placed them. So two different ways we've tried doing this. One has been to cage sort of this environment. So put a sort of 6 foot by 6 foot by 6 foot mesh cage over this release point, which helps contain and constrain the area that you're going to go back to monitor for eggs and for damage in the next year. But it is more logistically involved. We've also tried some open pupil releases. So that's what you're seeing in the middle right, are just containers that have essentially been staked out in the middle of one of these Phragmites patches. So the pupae, the adult moths can emerge and sort of go wherever they will. We're sort of hoping that they'll be able to find each other and stay close together. But again, that's one of these comparisons, is to figure out what works best. So 2021, as I mentioned, was a really encouraging year for us in terms of the amount we were able to do. It was the first year that we really had substantive releases across several locations. So in 2021, we were able to do 7 new release sites, and we released approximately 6,200 insects in various life stages. So a bunch as eggs, as I said, many as larvae. This was the sort of dominant form of that. A handful of pupae, because they are quite a bit more work and time to produce. And then a few moths, although that was more just pupae that they emerged early on us when we were putting those cages out. So the emphasis in this year, again, thinking to those research objectives, is really on, we're trying to get insects out, but primarily we're trying to figure out different release methods and timing. How do we do this? What's the right time to do it? Which methods work? And then we can scale up from there. So I want to devote a bit of time here to talk about preliminary monitoring, sort of what we've found so far. If anyone's caught any of our talks in previous, a lot of this will be sort of familiar up to this point. An exciting thing this year is that we've been able to actually go back into things that we've released in the spring as larvae anyway, and try and monitor for damage from those larvae to see whether they've been able to start feeding in these populations. So monitoring in general is an essential part of this whole process post-release to understand where they are, how they're dispersing, the impacts that they're having, and so on. So I'm going to walk us through our emerging protocol, because again, we're learning how to do this, for stem inoculated larvae. So these would be those larvae that are put into those blocks of cut stems. So these are those blocks again. So you can see them in the lab. They've been prepared in blocks of 50 here. So again, each of those has 3 larvae in them. So 150 larvae per block. And I will say again, just one of those blocks takes quite a bit of time and effort to put together. These typically go out in early May. We're trying to target, and this is a theme that'll come up later as well, trying to align the phenology of the insect development with the phenology of the plant. So trying to get those timings to line up properly. With the larvae, we're trying to hit a period when Phragmites stems in the field are emerging from the ground to a height of about 30 to 60 centimeters. So once those stems are out there, another really nice thing about this particular method is that we can actually go back and recollect the stems that we put out to try and assess that initial introduction of larvae into the field. And that's something that we can quantify, which is really useful. So after about 2 weeks, any larvae that were in there should have left by now. And when they leave behind some of those larval boreholes. They're harder to see in this image here, but I've kind of circled some of them there. They're these little kind of pinpoints in the leaf sheath. So we can bring those back in, we can quantify it and try and gauge how many larvae made it into the patch. So that would happen in sort of mid-late May. One of the big things we've been trying to learn to do this year is how do we actually look for damage? And what's been emerging as a really nice time to do this is to go back and look in mid-June. So this is a period when the damage is fairly obvious. So the larvae have had enough time to feed and do some damage. The Phragmites is tall enough that you can see that, but not so tall and not so surrounded by other vegetation that you can't find these damaged stems. So this is some monitoring that we've done around the Lake Erie area. So there'd be a release point at that PVC post in the middle there. There's a hula hoop that we use for monitoring is actually really useful for that, sort of in that immediate area around the release point. And then we've got a 2-meter ruler there. And what we use are these little hair clips that I've glued flagging tape to. So whenever we see a damaged stem, we can kind of tag the stem and then come back later and measure various parameters of it if we choose. So we want to look for insect damage. We also want to start looking for insect impact on the population. Now, because we're looking at sites where the larvae just went out this spring, we're not really expecting to find any kind of impact on the whole population per se. This is more just about what are they doing in the immediate area around the release point as evidence that they are present and that they are feeding. So a good time to do this seems to be early fall. So this is a point when Phragmites is starting to senesce. Most of the other vegetation has died back a bit. And this can be a good time to go in and measure things like stem density, stem height, panicle development on the stems, and so on. So from that 2021 monitoring of some of these larval release sites, again, this would just be the first window of time in which we have monitored them post-release. Of course, the gold standard for knowing if they're there is to actually find them. So in this case, we'd be looking for larvae. And so you can go in and you can open up stems that seem to have damage. And that's where we got this photo of that Lenisageminipuncta larvae sort of happily munching away. So that was really exciting to find. One important thing about these early sites is we are trying to manage and balance monitoring and learning how they are spreading and the impacts that they have with actually getting them established on site as well. So we try to exercise a fairly light hand in monitoring so that we don't disrupt this population that we're trying to establish. So more often than looking for the larvae themselves, we end up looking indirectly for other evidence of their presence. So that's why things like the emergence holes in those inoculated stems are a really nice indirect measure that we can quantify. Those larval boreholes that we showed earlier of them entering and exiting stems are also fairly diagnostic. Again, they're not always this obvious. These are sort of the later instar, larger larvae leaving these behind, but they can be a good thing to look for. When they're feeding in the stem, I mentioned they can cause stem wilt, stem mortality. On older stems, the stems don't tend to die, but they will sort of stunt the stems in various ways. So that feeding in the upper internodes of the stem will stunt development above sort of at that growing point. And the plant will often respond by putting out secondary shoots, and you can see one there where the arrow is sort of a thinner side shoot that comes off the side. So when we see a borehole and that branching stem, that's a good clue that something's going on there as well. There is some stem wilt and stem death, again, especially earlier in the season on the younger stems. When they're fed on, they're more likely to die off. I've learned that this is incredibly difficult to get a convincing photograph of. It's much more obvious when you see it in the field, but these are some of my better attempts at it. So you can see here, these are stems where the borehole has been circled there. And then looking above it, you can see past the growing point there, the leaves and sort of the stem above it tend to wilt and eventually sort of die off. So again, it's this combination of the borehole and the other damage that really tips us off. So this has been really exciting to see. We also want to throw a bit of a caution on this. This is really good to see, but at the same time, we want to remind everybody that this is that first year since they went out. So these would just be larvae that we put out in the spring. It tells us that they have managed to molt and survive through the year and have some feeding damage that we can detect, which is great. But what we really want to be able to see is, are they able to reproduce and overwinter? So produce, lay eggs that survive the winter and will hatch at the right time the following spring. So this is going to be one of those really critical windows. So this spring going forward, looking to see whether that population can establish between those 2 years and become self-reproducing. Past that period, of course, we want to see if there is an opportunity for long-term persistence, so monitoring from year 2 onwards. And again, emphasizing that this feeding damage is really great to see, but that population level impacts are going to take time. So that's something we'll be looking for and sort of hoping to see in that second year going forward and sort of increasing over time as well. But it is a really good starting point. So a few key lessons I wanted to pull out from this year of work in particular. A few themes have kind of emerged for us that we're going to be paying attention to going forward. And one of the big ones is that release timing really matters. As I mentioned, one of our big priorities is to try and align the timing of the insect life cycle with the phenology and development of the plant, of introduced Phragmites in this case. So this figure sort of shows that somewhat indirectly. We have some dates along the horizontal axis. Along the vertical axis, we have stem height of emerging Phragmites. And each of these points is a different release site where we released insects in some stage and what the average height of the Phragmites on site was at the time. And I mentioned before with the larvae, we have reason to believe we want to target this 30 to 60 centimeter height, and that only occurs at specific points in the year. So we've seen when we release at sites that has Phragmites stems that are very close to that target, those are the sites where we tend to see the most feeding. When we look at and compare to a site where we've maybe released later because we wanted to compare it or we had to release at that point, when the Phragmites is quite a bit taller, it's tougher, it's harder for the larvae to get into those stems. We see less feeding later on in the season. So trying to hit that window is going to be really important. We also saw from that very first 2020 egg release and then in later work as well that predation is likely going to be a limiting factor on the establishment of these populations. That's not surprising for this kind of system. So this could be predation of different life stages, the eggs, the larvae, the pupae, potentially the moths as well. So because we are in the business of trying to grow these populations, we want to try and find ways to limit that predation early on. So this could be through physical means like our screened cup to try and offer some measure of protection to the eggs. It could also factor into timing, so trying to release material at the correct point in the season such that it's not sitting out there vulnerable to predation for longer than it needs to be. Monitoring is essential through all of this, and figuring out when the best time to do that is has been important. And we have this emerging model of going in the midsummer to look for feeding, and then going in the early or mid-fall to look for impact. We're always looking for new methods or refinements on methods that we've been developing. So I mentioned that stem sort of larval inoculation method was working really well, but is a lot of work. So to give you a better idea of what that looks like, if we sort of start on the left of this, you have a shot of this is a small petri dish that has 1,000 eggs of Lenisageminipuncta in there. These are very sort of fiddly, kind of clingy eggs that are difficult to work with, and they will hatch out into tiny, clingy, difficult to work with little larvae that you can see in the next slide there. So we then need to take 3 of those without having all the rest run across the whole lab and get them into these stems that we've harvested at the right time and stored in the lab. And so again, making these blocks of stems is quite a bit of work, but they do work well. So we're going to keep using this, but we're also going to be looking for refinements of this and other options as well. So to give a quick look at some of the upcoming work that builds off of some of these themes and some of what we've been seeing so far, of course, we're going to be monitoring all the releases that we've done so far, looking for that overwintering period in particular, and to assess, again, feeding and impact on the populations. We're really interested in cycling back to looking at egg releases again, as we did in 2020 there, because this is a really practical life stage to work at in many ways if we can get this to work. So we've set up a refinement of these protected containers that have this mesh bottom on them. And we're doing a comparison here where we look at whether it's better to put out eggs before the winter so that they sit on a site in winter, they're ready to go, they experience natural temperatures and everything, and then they just hatch in the spring. But are potentially vulnerable for a longer period of time, Or is it better to run them out in the spring just before they're about to hatch? So we have a few sites set up for this that we'll be comparing this year. We're also getting ready to do a larger scale release this year, and then a few others in upcoming years as well, to look at impact and dispersal. So we have some really nice sites with larger populations and sort of satellite patches of Phragmites, so that we can look at how insects disperse between populations or patches over time. And we're very excited to be working with Dr. Rebecca Rooney and her team out of the University of Waterloo. They've done some great work at vegetation monitoring following Phragmites management. So we're really excited about that collaboration. We also do have two biocontrol agents, and often we sort of have talked about them sort of as one effectively, but they are a little bit different. So we'll also be doing a site this year to compare the two different species in terms of rate of spread, what their damage looks like, and how their impacts combine as well. We'll also be doing some additional rearing so that we can produce pupae, moths, and eggs for various other experiments. Looking even further afield to 2023, again, monitoring through all of this. And as time goes on and we know which release methods work the best and so on, there'll be an increasing emphasis on what happens to the vegetation. So what is this doing to the Phragmites, and what do we see in terms of recovery from other desirable vegetation on these sites? And again, working with Rebecca's team on this will be really useful in that regard. Once we get all these methods down, it's then just a question of scale. So scaling up our ability to produce insects, to rear them, and to release them across a larger geographic scale on the landscape. And so to conclude, I wanted to throw in just a few general reminders about biocontrol. So these would apply to our Phragmites system here, but also just generally. And the first is, as we've emphasized, biocontrol is a long-term solution for large-scale problems, and Phragmites is a large-scale problem, and it needs a long-term solution. So this is actually a really nice fit. What we're trying to go for is not complete eradication. Complete eradication can have a number of problems related to it in terms of, for example, the possibility of reinvasion by Phragmites or secondary invasion by other species. What we're trying to achieve here, if we think to that early graph of the biocontrol agent and the weed populations, is about sort of gradual suppression over time. And this can be combined with other management, or it may be combined with biotic resistance from a recovering native vegetation community that is just able to establish itself and coexist. When we talk about biocontrol, we get a lot of discussion about risk, and this is a really important part of the discussion. One thing to remember here is that when we're talking about biocontrol risks, these need to be compared against the risks of other management. So any tool will have risks associated with it, including things that we're already using. And also doing nothing has perhaps the greatest risks in many cases. So in places where it's not practical, or there isn't the money or what have you to do anything, we know what happens when introduced Phragmites is able to just run rampant. And risk is really at the forefront of that whole biocontrol program development. When we talked about those stages and those steps, the amount of time and effort that goes into finding agents that are highly specific to the weed, sort of a lot of work goes into this particular consideration. And finally, while we think biocontrol has really a lot to offer here and is a useful tool here, it's also not a panacea. This is not about fixing everything all of a sudden on its own. It's about supplementing the other options that we have as another tool in the toolbox. So thinking about that timeline, this program has been in development for over 20 years. This takes time, this takes a lot of work and a lot of people. But as of 2021 now, we have multiple releases in Canada at least, which has been really exciting to see and a neat time to be looking at it. Biocontrol does take time, and we do need long-term monitoring to really get a clear picture of what's going on. But it's also really encouraging to see the evidence that we've seen so far of that initial insect feeding. So we know we can put them out into the population. We know they can feed through a number of different molts and do some damage. So this has been a really encouraging start. And as we go forward with this, there will be, of course, ongoing work for years focusing on monitoring, refining the methods, and all building up to this increase in the scale at which we're able to rear insects and release them across the landscape. So hopefully we're going to be seeing more of this and hearing more about this in years to come, so do stay tuned for that. And as we sort of alluded to, well, as we sort of directly said early on with the slide talking about the international nature of this work and the scale of this work, really this doesn't happen without the contributions and efforts of many individuals and many institutions. So of course, we're very grateful to those many different collaborators on both sides of the border there. So thank you very much for that, and happy to take some questions if we've got time.
Thank you so much. Yeah, we'll start going into questions.
Did you want to, someone going to read them off, or how do we want to
Yes,
handle this?
I will read
Okay,
them off. I'm
perfect.
just opening up the chat. There's
Okay,
a lot
sounds good.
already.
Oh, I'm sure. That's great.
Okay, so our first question, how do adult moths leave the stem? I'm assuming they do not have mouth parts capable of chewing. Does the larva chew a hole first before pupating? Seems like having an open hole like that would invite trouble.
Right. So sort of I haven't observed this directly, but my understanding of the biology would be that they do prepare that exit in advance, sort of prior to that pupation stage, such that it's not fully open, but it is slightly covered. So that's a surface that they're able then to push their way out of once they do emerge. So they don't require those specialized parts to be able to do that, but they do have some measure of protection as well.
Great. We have a question from Robert. Tangentially speaking, what is the current best practices for differentiating between the native and non-native Phragmites?
Yeah, so that's a really interesting one. And I think as we've gone through this work, we've seen over the years sort of a shifting interest and awareness, and I think generally for the better, about the presence of this native subspecies. We've seen situations where you even have native Phragmites populations that are being managed, they're being mowed or being sprayed just because sort of no one thought that it was there or was able to tell them apart. There are situations where it's, I would say, very obvious, and some situations where it's a little bit more cryptic. Usually what you'll hear is sort of it's sort of a suite of different characteristics that in combination make them different. What I think can be overemphasized is kind of the size and density of the patches. Certainly on average, our introduced Phragmites tends to be a lot more dense, it's taller, you have much larger panicles and so on. But you can also get very weedy introduced Phragmites patches, and you can get very robust native patches. So I sort of tend not to prefer those attributes. We're working on a paper right now. We have colleagues at Agriculture and Agri-Food Canada. We're working with Tyler Smith in particular, doing some molecular work. So from that background survey that I mentioned, we have introduced the native populations, and we know what they are from the genetic work on that, and then we're lining it up with the morphological traits and seeing what best defines them. The one that has emerged for us that we haven't seen talked about as much elsewhere is actually leaf sheath retention on standing dead stems. So Phragmites, as it senesces, a lot of those stems will actually stay standing in the patch. And on those dead stems, the introduced subspecies tends to retain the vast majority of its leaf sheaths. So they'll still be stuck to the dead stem, whereas the native will drop most of those. And this trait is really a nice sort of clear one. It's one that you can see sort of year round for the most part. And so that's one of the better ones that we've been seeing. So we're hoping to put that work out soon, and would be happy to share that once it is out in the world. I would also caution this tends to be somewhat regional. So we're seeing that depending on where you are, different traits may vary between populations. So our work is kind of Southern Ontario specific. That may be more or less relevant depending on where you are.
Yeah, great, thank you. We have a next question from Ron. Is there any chance that the moss will evolve the ability to eat native plants over time?
Right, of course. And this is an important question and an important consideration, and it all comes down to these sort of questions of risk. You can't say, for sure that there is across a long enough time scale. There's of course not. You can't say for sure there isn't a possibility of this, but so much effort does go into ensuring that right now these species are highly specific, and that's what we see, and they don't get through that petitioning process without having that high level of confidence in the security of that. So they are very specific to that, and the risks with what might happen over a longer period of time, again, are sort of weighed against the risks of the other tools that we are using right now, very non-specific tools, for example, or situations where we can't do anything at all to deal with that. So it is at the forefront of the discussion, and it is something that we feel very comfortable in this being a very safe tool to use here.
Thank you. So we have another question. Have you studied what would happen if the larvae ran out of invasive Phragmites to eat? Would their population collapse, or would they move to a less preferred species?
If they ran out, right. So this sort of gets down to the host specificity sort of question again. Due to a number of different behavioral and physiological constraints, again, they are very specific to this species. So what we would tend to see is they wouldn't be able to move into another host. So if we think of that example diagram of the weed and agent populations early on, what we see is, and what we'd expect to see and hope to see, is as the target population goes down, over time you also get a sort of cycling of the agent population that also decreases. As they have less food, the population becomes lower until ideally they both coexist at sort of a lower level. So longer term, that's what we would expect to see, is low density Phragmites, low density Archanara as well on the site.
Okay, great. We have another question from Steve. How do you see succession taking place in large stands that are controlled by moths?
Yeah, so I'm going to say we don't know yet, but we're certainly curious about it. This is where a lot of this work that we're planning with Rebecca Rooney and her group. So our big release coming up this year, and then sort of planned for the following year or two as well, are focused on this question primarily. So once you start to dial back your Phragmites dominance, what comes back in place of that in terms of vegetation community? And this is a really interesting, really important question. I would say from other work that their group has done, looking at other control tools like herbicides, for example, combined with mowing and whatnot, a lot of this will depend on what other vegetation is present on site, what can move onto the site, what is present in the seed bank in particular. So whatever species you have present, their ability to reestablish themselves once you have openings in these formerly highly dense sort of dominant populations of Phragmites. So kind of what's in the seed bank is one of the big questions. There's also the potential, of course, we're not doing this right now, but with any control option, the possibility of active restoration, so going in and planting or seeding or doing what you will to try and sort of structure that successional trajectory a bit as well, is also an option to consider to help that along.
All right, thank you. Another question from Angie. We have, how are you differentiating success between the two species in your study? Are they being released separately?
Yeah, so far they have been. This is both intentional to keep them separate at this point, but also because we often have different supplies of different species year to year, sort of from rearing efforts to do that. This year, coming up at one of our sites is going to be the main attempt to sort of focus on that. Keeping them separate. Basically, we'll be manipulating different patches where we can have one that is for each of them and sort of another where we can put them together. One of the nice things is that in the first year when you release them, the larvae don't go very far because they're not very fast. So if you put a few points, even sort of a few meters apart, you can have a fairly localized area that they're not going to disperse out of. So you can keep them separate. After that, of course, once they have the ability to fly as moths, then it's sort of an open game and they can kind of go wherever. They are morphologically distinct as larvae and as adults. So, if you're seeing damage, you could open that stem up if you wanted to and see who's actually in there to try and figure it out. So we have a few ideas how to compare those.
All right, thank you. Another question, we have a question from Colin. Are there any other species, native or non-native, that that create boreholes in Phragmites, either Phragmites Australis or Phragmites Australis americana?
Yes, yeah. So this is an an important thing to consider as well, is that this won't be the only insect that is occurring on Phragmites individuals in the field. So we've members of our group, Rhoda De Jong, who was a postdoc who was in this lab before myself. I was involved in some of that work later on as well. She actually took some of those sites that we looked at along that background survey area and was comparing insect communities in both of the subspecies, looking at how they co-occur, what is there. And one of the important things to look for is what kind of damage they can produce. So far, the boreholes seem to be fairly distinctive. Again, certainly for the later instar larvae. When they're fresh, they're pretty clear. When they get older, they can look a little bit more like other things. Other clues as well, like that secondary branching that I showed, there are other species of insect that can cause similar damage. Various Liparis species can do that. And so we're going to be looking at whether that architecture does indeed look diagnostically different, or if you need to combine it with other cues, like in our case, we were primarily only recording something if it co-occurred with one of those boreholes that we were pretty sure that's what it was. So it was sort of the weight of evidence of different characteristics together was what allowed us to say this is what we think we're seeing.
Great. And we have a few questions that are kind of similar about dispersal. So I'll kind of ask them side by side. So along with the dispersal questions, how far can each of the moths fly? And we have another, does anyone have a ballpark idea of how far these moths might spread on their own per year?
Right. Very important questions, and we don't really know at this point. That is going to be, now that we have insects, hopefully, I mean, we're hoping that we'll have them established soon. That's something we're going to be monitoring for once we have moths that are able to spread, monitoring for that dispersal, sort of how far, how quickly, all of these will be questions. So one of our big study sites that we're setting up this year is going to be targeted for that. So essentially releasing into a mosaic of patches of Phragmites with sort of space between them and monitoring these kind of satellite populations to see how they move between them. That will be, of course, very important for understanding the longer term impact and how quickly we can expect them to move across the landscape, what's sort of too far a gap for them to bridge, and so on.
Okay, great. Thank you. That seems to be the questions that I see. I guess, Sam, if you see any more that I missed, please let me know.
And I would say if people want to get in touch as well, I did have my contact information there, so happily to follow up separately if there's anything people didn't have a chance to get to as well.
Yes, thank you. So on this slide, please see Michael's contact information if you have any questions about his work. We want to thank you again for presenting today.
Oh, thank you very much.
Yeah, so just so you guys know, the recording of today's webinar will be shared through the Eventbrite. You'll get an email and you can view it and on our website by the next week or so. And we do have an upcoming webinar for March 23rd at 10:30 a.m. Eastern. We will have Amy Corazino-Lyon of University of Wisconsin Green Bay and Brian Galensky of Ducks Unlimited, and they will be presenting A Tale of Two Species, Restoring Native Wild Rice Through Non-Native Phragmites Management in Green Bay Coastal Wetlands. So please come back to the GLPC's website and register for that one next month. And I want to acknowledge the U.S. Geological Survey and the Great Lakes Restoration Initiative for providing funding and support for the collaborative. As a reminder, the webinar recordings will be posted on our Great Lakes Phragmites Collaborative website in the next few days. And please, if you have any additional follow-up questions or comments, you can reach out to us at phragmites@glc.org or reach out to Michael at his email. And thank you all for joining us today.
Yep. Thanks again for organizing and everyone for coming out as well.
Yes, of course.
Okay, take care.